All right, y'all. Testing. This video, but the microphone was muted, so we're gonna do it again. Cause that's just what it is, brother. It's just what it is. All right. This video is gonna be about interphase and mitosis. Interphase is the stage in between replicated divisions, also known as mitosis, which is cell division and replication. So, what kind of cells undergo mitosis? Labile or proliferative cells. These cells continually undergo regularly scheduled mitosis. Think of epithelial layer skin cells. Your cells in your skin are constantly replicating and pushing those older cells to the top, and the ones on the very top are dead. So they have to constantly get renewed. You have gastrointestinal tract cells, GI tract, so your gut. Got all that hydrochloric acid, that gastric juice. And it's probably going to need more cells to, replace those damaged ones. You got the urinary tract cells for urea, and the cells exposed to that. You gotta keep on replacing those. You got hematopoietic stem cells and red bone marrow, so you know that your red blood cells are created in your bone marrow, your red bone marrow. And when they break down after wear and tear, then they break down into some proteins like bilirubin and other serum proteins, but like iron. But that's after they break down. But you need to replace those cells so that your body can get oxygen. Those cells are known as labile or proliferative cells that chronically undergo mitosis, consistently and continually undergo regularly scheduled mitosis. Next, we have stable cells. These cells don't replicate unless they're signaled to by a strong stimulating factor. An example is the liver cells, which can regrow up to 40% if they undergo cirrhosis or liver scarring, tissue scarring. You have epithelial cells of the kidney. You got the proximal tubule, you got the distal tubule, you got the loop of Henle, you got the convoluted tubule. Those cells, those epithelial cells, can undergo mitosis as well. It's stimulated by external factors. Excuse me. So then we have another form of stable cell, which is the alveolar cells of the lungs. I didn't think that these cells could undergo mitosis because that's why I thought like lung cancer was so bad. But honestly, it makes sense because lung cancer, right? Uncontrolled proliferation of the lung cells. I don't know if that has to do specifically with the alveoli, but I thought the alveoli were unable to be replicated or undergo mitosis, but apparently they can. So there you go. But then you probably need a strong stimulating factor to initiate that process. And then you have the permanent or amitotic cells. These cells don't undergo mitosis. So you have nerve tissues. That's why, muscular sclerosis is a thing. You get that degeneration of the myelin sheath surrounding the neuron, leading to neuron damage. Those neurons can't be replaced. Those nerves, not neurons, nerves. Those nerve tissues. You got skeletal muscles. So like if you get, muscular dystrophy, got that dystrophin buildup. Those skeletal muscle cells cannot replace themselves. But this is interesting because I want to know like how when you like lifting weights, how you're tearing tissue, or when you're going on a long run, you're tearing tissue, and that tissue comes back stronger. I want to know how that process goes. If skeletal muscles are amitotic, how are the cells? How are you, gaining muscle mass? How's that tissue rebuilding itself? I think it's repairing. I don't think it's undergoing mitosis. But like they say, you can only get a certain amount of muscle tissue, and then after that, it's just like repairing itself. But I'm not sure. I have to research it, which will be interesting. Then you have cardiac muscles. So if you get scar tissue in your muscle in your heart, it's pretty bad. You can get risk for heart attack, embolisms, another thing like that. Some thromboses, blockages, mostly embolisms, and aneurysms. So you can get a whole bunch of bad things, but cardiac muscle can undergo mitosis. So Before we undergo cell replication of this mitosis, going from, we have here one cell into two cells, two sets of chromosomes, 23, n equals 23. Get 23 from your mom, maternal, 23 from your dad, paternal. Slap them together, you got 2n, and we need both of these daughter cells to have 2n as well. So how does that happen? So before you undergo mitosis, you have something called interphase. Interphase is a series of transitionary states of the cell that occurs prior to mitosis. These series of phases are the G1, GAP1 phase, the synthesis phase, the S phase, and the G2 or GAP2 phase. You also have this G0, G0 or G0 phase, and that's a non-replicative state where cells usually remain until provoked by stimulating factors. So it's kind of like a detour. So yeah, G1, G2 or S, G2, mitosis, and then you can branch off to G0, or you can go straight back into G1. Excuse me. So what is G1? G1 or gap phase, GAP1 phase. Most cells exist in this phase, just because they're just existing. And in G1 phase, you're replicating cell organelles in the parental cell. So you're increasing the number of organelles so that you can get ready for cell division, because you need to make sure that before you divide into two cells, you need to make sure the first cell has everything it needs to become two cells. So that's interesting. Second, after you make sure you have all the organelles necessary for cell division, you're going to synthesize proteins and enzymes required for DNA replication, such as DNA polymerase 1, DNA polymerase 3, primase, helicase, topoisomerase, all the enzymes you need to replicate all the DNA in the parent cell. If you've watched my replication video, you know a little bit about that. I'm not going to elaborate more here. The last part of G1 phase is that you're going to repair thymine dimers that exist in DNA. So thymidine dimers, thymidine exist in DNA. That's where DNA damage is, and you're going to repair those through a series of mechanisms. I know that in double strand breaks, you have something called ataxia telangiectasia mutated gene, which is a gene that helps transcribe and repair those double strand breaks, or perhaps thymine dimers as well. And the whole point of this is that in the G1 phase, you're proofreading the original DNA. Before you go into the synthesis phase, which is the next phase, you're going to want to make sure the DNA you're synthesizing has a correct template to use. So you're going to replicate all the organelles in the cell. You're going to synthesize the proteins that you need for replication, or for DNA replication, and then you're going to repair the DNA damage in the original DNA. So next you have the synthesis phase, the S phase. In the synthesis phase, this is DNA replication. A whole video on this, but the main takeaways are it's bidirectional. So you have the leading strand and the lagging strand that occurs in the 5 to 3 direction following the helicase. And then you're going to do that until you have 4N. So we already established that N is 23 in humans. So you have 223, 2 times N, 23 from your mom, maternal, 23 from your father, paternal, and then you're going to have double that, so 4N before the cell divides. And the DNA polymerases 1 and 3 begin synthesizing the new strands of DNA using the parental strands. Watch the replication video if you want more information on this process. After you have completed the synthesis phase and successfully replicated 2N DNA and achieved 4N DNA, and in the G1 phase, you've replicated the organelles, you've synthesized the proteins required for DNA polymerization, and you repaired the DNA damage. You've synthesized the DNA. Now you're in the G2 phase. The cell is literally and physically going to grow. It's going to get bigger because now that it has all the things required inside, it's got the DNA and the organelles. It needs to make sure it has the space to put all that new stuff. So the GAP2 phase is the cell growth phase. It's going to increase the cytoplasm to ensure it's enough for 2 cells and not just one. Ataxia telangiectasia mutated is a gene. It's a master regulator of double-strand break signaling and stress responses. So if there's any damage, this checkpoint is going to halt. It's going to phosphorylate P53, a tumor suppressor. It's going to activate that. And P53 has a bunch of pathways that we will go into detail in the next video, which is going to be very detailed. So just be ready. Warning you now. To either initiate apoptosis or activate DNA repair enzymes. So it's either going to repair the DNA using some enzymes, or it's going to throw the whole cell away through apoptosis or programmed cell death. So that's G2 phase. G2 phase is the growth. That's what you got to remember. It's about the growth, and you do a checkpoint. But we will get to that later. So after you've gone through interphase, you've gone through your G1 phase. You've made sure the organelles have been replicated. You've synthesized the proteins and enzymes required for DNA replication. You've checked the OG DNA for any mistakes in the G1 phase. Then you've gone through the S phase and you synthesized all the DNA. You've gone from 2N to 4N. You have enough DNA to undergo replication. It's all correct. And then in the G2 phase, you've expanded. There's enough space for mitosis to begin, and all the cytoplasm's there. The cell's big and healthy. It's ready to get going. Then mitosis begins. What is mitosis? Mitosis is a 4-phase process that results in mostly identical cell replication. After the cell cycle checkpoints have been cleared, the mitotic cell division process begins. This process occurs in eukaryotic cells that exhibit a phospholipid bilayer membrane, a nucleus with chromatin, and cytoplasm. And the 4 phases of this mitosis process are prophase, metaphase, anaphase, telophase, PMET. Take the first letter, PMET. So prophase. What is prophase? Prophase is when cyclin-dependent kinases phosphorylate lamins, histones, and histones. Yes. So lamins are structural proteins that hold together the nuclear envelope. They also aid with transcription, but you just need to know that the nucleus is going to disappear, like the nuclear membrane, the nuclear envelope, as they call it, is going to disappear, and cyclin-dependent kinases are phosphorylating lamins, these proteins that hold the structural integrity of these nuclear envelopes. When histones are phosphorylated, there's no data showing that it's a causation or relationship that histone phosphorylation leads to chromatin organization. But in one paper I was reading, it said that a histone over here was phosphorylated, and then the DNA over here started condensing. And it was like they can't show them in the same vicinity. And I imagine it's because nobody wants to dump money into that. There's no money into it, but it is what it is. So I don't want to get ahead of myself. Just say there's no reliable data to show this. It might just be a pure coincidence, but if you think about it, histones are positively charged. That's why they attract negatively charged phosphate backbones of DNA, get them to bind. You phosphorylate that histone, add a negative charge, neutralize the charge. The DNA might, move away from it and start condensing. We don't know, and nobody's studying it. So, or maybe somebody studied it. I mean, not. I wouldn't know. I've done enough research. And then you phosphorylate nucleases, which start just chopping away at that nuclear envelope. So these cyclin-dependent kinases have a pretty big role in prophase, and they activate these lamins, or they phosphorylate these lamins, degrading those. They phosphorylate these histones, which might lead to chromatin condensed chromatin formation, nucleases, so they can start chopping up the nuclear envelope, and they phosphorylate other nuclear envelope proteins to initiate mitosis. The chromatin condenses from euchromatin to heterochromatin, different chromatin. What's different from chromatin? Chromosomes. The nucleolus disappears. Next, the nucleolus disappears. The nucleolus is an apparatus in the center of the nucleus, which is where ribosomes are synthesized. Ribosomes consist of rRNA, tRNA, and ribosomal proteins. But the main thing you're going to want to know is rRNA. That's the big one. And ribosomes make proteins. So, the DNA for the protein making, ribosomes are in there. And ribosomes are groups of proteins and rRNA. So the nucleolus disappears, the nuclear envelope disappears, and microtubules made out of a protein known as tubulin. The microtubule centriole organization center. So centrioles are just groups of microtubules bound together. I think it's 9 of them, but don't quote me. It is late. Microtubule centriole organization centers form from the centrosome. So there are these things called the microtubule centriole organization centers, and they form from something called the centrosome. And then from these microtubule centriole organization centers, you have polar and astro microtubules. Astro microtubules point away, polar cells point towards the chromosomes, but we'll get to that later. So that's prophase. Here's the nucleus, here's the cell, boom, go through prophase, nuclear envelope disappears, the nucleolus disappears, which could be marked by a dot here. Nucleolus, yellow, really? Okay. Hello? Oh, that's hard on that. The nucleolus disappears, and the microtubule centriole organization center forms. And there's 2 of them because you're going to have microtubules on both sides. So that's important. So after prophase, the nuclear envelope's gone, the microtubule organization centers formed. You're going to have metaphase. And metaphase, if you look over here, is where the chromosomes line up on the center. So let's show you what chromosomes look like. So chromosomes are these loop-de-loop things. You're going to have 1 loop and 2 loop, because remember, we have 4N. So each chromosome actually has 2 chromosomes worth of DNA. And they're connected through this thing called the centromere. And a chromosome is defined as having 1 centromere, right, by the number of centromeres. So there's 1 centromere here. It's notated by the purple. So this is 1 chromosome. This is all 1 chromosome. However, these 2 short arms and long arms are called sister chromatids. And that's what's going to separate in mitosis. Mitosis, sisters, mito. You think of mitosis, think of mitosis. So they separate. I don't know. You see this stuff enough, it just you start to come up with ways to remember it. But you think of that, your ptosis and the sister chromatids separate. So how do they separate? Well, Remember those microtubule organization centers that we were talking about and how they migrated to both sides of the cell? So these things right here, these things, they migrated to right here and right here, right? See those red dots? And they're microtubules. We have the polar ones reaching towards the center. Remember, and the astro ones. Let's draw the astro ones real quick, just so that we keep the continuity here. So you're gonna have some astro ones just tailing off the back here, just in their own land. Probably rooting these microtubule organization centers to the cell, but did not look into the function there. In those microtubules, the polar ones are going to latch onto something called the kinetochore on either side of the centromere. So the centromere is the center of the chromosome, right? Well, they have these nice proteins called kinetochore that are on the outside of the centromere. And they're kind of like sandwiching the centromere. And on those kinetochore, you have a bunch of proteins. You have securin, you have cohesin, and you have separase. But I'll get to that in the next one. Anaphase once they start to separate. So just so I make sure I read this, the metaphase happens when the chromosomes line up at the metaphase plate. That's the first thing. Then those microtubule organization centers migrate to the poles of the cell, where those microtubules then connect to the kinetochore on either side of the centromere, and they get ready to pull. And between here, we have the M checkpoint. So we got to make sure, because a lot happened there. You have them line up, you have the microtubules latch on to those kinetochores. You have to make sure that's all going correctly. And there is a very interesting checkpoint that occurs there. So what happens after your microtubules and microtubule organization centers have migrated to the opposite poles? The microtubules have latched on. The polar microtubules have latched onto the kinetochore. And the chromosomes are ready to be separated. So motor proteins such as kinesin and dynein pull the chromosomes to opposite sides of the cell using the mitotic spindle or microtubules, or the microtubules. So you've ever seen kinesin and dynein, you can see they're like little pulley proteins, and they pull the load, and the load in this case is chromosomes. So as I was mentioning earlier, here we have something called the anaphase promoting complex. So after metaphase, after the chromosomes line up at the center and they're ready to be separated, after they pass through the M checkpoint, you have the anaphase promoting complex, which comes and binds to the securin protein, which was holding that centromere together with those foreign chromosomes. Those extra loaded chromosomes. Gotta be careful with my English here. So that anaphase promoting complex degrades or inhibits that securin. Once that securin is inhibited, separase, the thing it was inhibiting, so securin makes sure separase can't get its money. Separase is a pair of scissors. It's going to cleave cohesin, which is keeping the centromere together. Once cohesin is separated, the motor proteins, kinesin and dynein, can start pulling those chromosomes away from the centromere and towards the poles with those microtubule organization centers. And that's anaphase. It's when it's pulling. Metaphase is when they're at the center. Anaphase is not when you see the microtubules. Anaphase is when they're pulling away. So it's actually separate. It's going to look something like this. I remember I got a test question wrong because I saw the mitotic spindle and it looked like they were pulling, but they weren't separated yet. And I got that question wrong because anaphase starts once there is space between the metaphase plate and these separated sister chromatids, um, chromosomes. So there's a nugget if you want it. Wise man learns from the mistakes of others. There you go. So sister chromatids separate when the anaphase promoting complex inhibits securin. When securin is inhibited, separase is uninhibited. It binds when securin binds to the anaphase promoting complex, it's no longer bound to the separase, and that means separase can begin to degrade the cohesin proteins, making the centromere able to be separated by the kinesin and the dynein motor proteins, and that separates the chromosomes to each side of the cell, or the two future cells. So lastly, after we have anaphase, we have telophase. And telophase is the final phase. So after the chromosomes have been pulled to the side of the cell, they now have equal amounts of cytoplasm. They have equal amounts of organelles. They have equal amounts of chromosomes. They both have 2N, enough to become fully sufficient cells. They form the cleavage furrow. The cleavage furrow is formed from myosin and actin proteins contracting, resisting tension, and the structure, the infrastructure of the cell starts to come to fruition. The nuclear envelope reforms. Remember those lamins? They start to assert themselves, and that's telophase. At the very end of telophase phase, you have cytokinesis, and cytokinesis is when the cytoplasm moves. So they slice that together and become two separate entities. That is cytokinesis. So that orange line represents cytokinesis, like the full separation of two separate bodies. Telophase is marcated by the cleavage furrow, which is this little pinch here. See, pinched. And then cytokinesis is the one that actually separates. But it's all deemed telophase, but that's just nuance, which is important, but it's nuance. So, after the telophase and cytokinesis, the cells undergo the G0 phase that we were mentioning earlier. So, as you know, we have the G1 phase where they replicate the organelles, they check the DNA for any errors, start synthesizing those DNA replication proteins and enzymes, polymerase 1, primase, topoisomerase, helicase, etc. Then you go to the S phase, you start synthesizing that DNA. You need 4N. You need to double that DNA. Go through the replication cycle enough times. Then, after that, you get to G2. Is the cell big enough to actually handle replication? Do we have enough cytoplasm? Is the cell healthy enough? Then it goes through mitosis. After mitosis, at a phase, you have the M checkpoint. We'll go through all the checkpoints in the next video, but there's way too much to put into one video. I might actually separate into a couple videos. Just go through each of the pathways. Because then I can go in depth. But I don't want this page to be about, cell cycle. But it'll be what it needs to be, right? I really want to get to papers, but we have to do the due diligence. So the G0 phase, the G0 phase. Telophase is followed by the G0 phase, G0 phase. It's a quiescent phase. So it's a resting phase, and it requires a stimulus to get out of it. So like I mentioned, it goes through G1S, G2M, then it can detour into G0, where it needs a stimulus, through a lightning bolt, like Shazam. Used to be Shazammed to get back into the cell cycle. Or it can stay in the G0 phase, and that's called cellular senescence. That's when the cell cycle is stable, and it has no plans of getting out of it. It's not quite amitotic because it might be able to go through mitosis, but unless otherwise provoked, it is senescent. It's a cellular senescent cell. It's Hey yo, that junk cut me off. But yeah, that was interphase and mitosis. Hopefully you found this video helpful. Let me know if you need any clarification or have any concerns or questions or critiques. Drop them down below. I'm happy to oblige. Make some changes. Still searching a format that's better than this because I don't know if this app is the best. Um, and also be ready for the next video. It's going to be a doozy. Probably going to use all this space for all these pathways. So it's going to be a lot, but I know we got it. Leave a like if you enjoyed, and we'll see you on the next one.